So bladder sensation. I will continue to make things even more complicated, I guess, in some way, and talk about is bladder sensation is always bladder specific, or maybe not. As you can see from the title, there is some crosstalk between the bladder and some of the pelvic organs. So changes in bladder sensation occur under different pathological conditions, including OAB, we know, there is urgency, and also there is additional pain component in IC or BPS. So nociceptive painful sensation in the bladder can result from altered sensation in neighboring pelvic organs, such as colon, uterus, prostate, due to a phenomenon known as pelvic organ crosssensitization. So what is that phenomenon? We believe that pelvic organ crosssensitization implies the transmission of noxious stimuli from a diseased pelvic organ to an adjacent normal structure, for example, urinary bladder, causing the occurrence of functional and very rarely structural changes, and specifically causing changes in bladder sensation. The studies from our laboratory and other labs showed that pelvic organ cross-sensitization may contribute to functional pelvic organ disorders such as painful bladder syndrome, irritable bowel syndrome, vulvodynia, and other functional chronic pelvic pain disorders. And this is just example on the right of significant overlap in these different conditions characterized by functional chronic pelvic pain. So, colon bladder crosstalk, what is the evidence? How do we think bladder sensation can be affected by some changes in the distal gut? So colon and bladder are functionally related, and this is what's been known for a while. Almost 100 years ago, it's been shown that micturition and defecation alternate under normal physiological conditions, and also studies showed that motility of the bladder could be inhibited by the distension of colorectum, suggesting anatomical and functional relationship between the bladder and the distal gut. Animal studies performed later on, and approximately they studied around 10 years ago, showed that when we cause inflammation or we apply some insult to the distal gut, that would cause changes in the bladder, such as increased frequency of contractions, reduced intercontraction intervals, and also altered micturition reflexes in animals with inflamed colon. So this is just examples showing the experimental animal models of colon bladder dysfunction. So what mechanisms underlie colon bladder crosstalk? We believe that there are different mechanisms, including neural, endocrine, paracrine, and immune. But the focus of my lab and what I would like to cover today, due to time limitations, mostly will be neural mechanisms of crosstalk. And this is just examples of different insults which could be applied to the distal gut, what changes they cause in the urinary bladder. Changes in contractility, release of neuropeptides, vascular permeability, detrusor overactivity, number of changes. The same actually is true when we cause some changes in the bladder. For example, chemical irritation, overexpression of MGF, neurogenic cystitis, inflammation, neonatal cystitis. All these changes applied to the bladder eventually affect colonic motility. So this crosstalk between the colon and bladder is bidirectional. So convergence and crosstalk, convergence in the nervous system and colon bladder crosstalk. What's the link between these two? So we believe that this crosstalk between the distal gut and the urinary bladder occurs mainly due to convergence in sensory innervation between these two organs. And interestingly, with each level in the peripheral and central nervous system, the number of convergent neurons increases significantly from 10-15% at the level of sensory ganglia to 30-35% of spinal cord neurons and even more in the brain. So there is significant amplification of convergent inputs in the CNS. So what is the evidence? Experiments in my lab, so we will start first from the sensory ganglia. Experiments done in my lab using retrograde tracers. One is, which is red fluorescent dye, was injected in the colon, and another one, which is blue, fast blue, was injected in the urinary bladder. So when we remove the sensory ganglia, this is just example of one of the cross sections of L6 level. We identified that the number of these neurons actually somehow received both dyes. And when we did analysis of, okay, somehow this is supposed to be different levels. So these are T13, L2, and this is L6, S1, S2. So L6, S1, S2 receive input from the pelvic nerve, and upper lumbar receive input from the hypogastric nerve. And you can see at all levels, convergent neurons are shown in brown. The number of these neurons is within a range of 10 to 20%, and it looks like L6 has more of convergent cells in comparison to other ganglia. So, okay, there is clearly anatomical convergence, but doesn't mean that the function of these neurons is changed. So we perform experiments when we isolated these convergent neurons, and you can clearly see that the same neuron has both dyes there, and we have patch pipettes. So we use patch clamp technique, put electrodes on these isolated neurons, and we characterized excitability, how excitability of these neurons changes when we have colonic inflammation. And this is just example of the tracers. This is control group. So when we induce inflammation in the distal gut, and this is transient inflammation. So this inflammation resides after 10, 14 days. But these changes are much more longer lasting in these neurons. And clearly there is decrease in the threshold, so these neurons become hyperexcitable. So what is the evidence that there is convergence also at the level of the spinal cord? For these studies, we used a different setup, and we used in vivo electrophysiology. We put balloons in the colon and in the bladder, and we used colonic and bladder distension to identify neurons in the spinal cord which respond either to one distension or another, or to both of them at the same time. This is just example of different spinal cord neurons which could be either excited or inhibited by the urinary bladder distension. And of course, it's stimulus dependent. This is red model, so up to 1.5 mils it would be noxious or physiological distension, and 2 mils would be noxious or painful distension. And as you can see, the number of neurons with excitatory responses was significantly increased in animals with inflamed colon. This is another example of the summary of this data. So these are responses of only excitatory spinal neurons, significantly elevated by colonic inflammation in overall population. These are convergent neurons, which is not surprising because convergent neurons received input from the inflamed colon. But interestingly is the graph in C. So these neurons definitely received input from the bladder, but none from the colon. And because this is in vivo system, we could confirm we used colonic distension, no response. So why these bladder-specific neurons become hyperexcitable at the level of the spinal cord? And we believe because of there is cross-sensitization. So the next level of convergence is specifically located, we believe, in the brain and in the brainstem. Excellent studies were done by Rita Valentina at the Children's Hospital in Philadelphia, and she specifically focused on the Barrington nucleus, which is pontine micturition center. So we wanted to know whether neurons in the pontine micturition center, which we know controls micturition, have any projections from the distal gut. And it turns out that yes, neurons in the, and this is single labeling. So on the left side, we see labeling from the colon. So Rita's lab had to use viruses, because viruses cross synapses to go all the way to the brain. And the right panel shows the bladder. So you can see in the middle, significant number of neurons in the pontine micturition center received input from the colon, and almost the same population received from the urinary bladder. Interestingly, in the Barrington nucleus, there are also two populations of neurons which received input only from the colon and also only from the bladder. The high magnification picture shows the yellow are convergent neurons there, and the summary of this study, up to 80% of neurons in the Barrington nucleus, which is pontine micturition center located in the dorsal pons of the brainstem, receive input not only from the bladder, which would be expected because it's pontine micturition center, but also from the distal gut. Also, small populations of these neurons receive exclusive input, either from the urinary bladder or from the distal gut. And they also did some functional studies in vivo in animals, and they showed when they use the same approach, either urinary bladder distension or colonic distension, they functionally characterized and showed that these neurons responded to both distensions. So there is clear not only anatomical convergence, but there is clear functional convergence in the brain as well. To summarize everything I said now, the colon and bladder are functionally related, with micturition and defecation alternating under normal physiological conditions. We believe that crosstalk between the colon and bladder is mainly coordinated by a convergence in afferent innervation at different levels in the peripheral and central nervous system, including sensory ganglia, spinal cord, as well as brainstem. Pathological changes in one of these organs may significantly affect and alter sensation in the neighboring organ, and also disrupt the normal coordination between the two organs. Altered sensation and pain observed in functional pelvic pain disorders may be coming from a neighboring pelvic structure rather than from the organ which we think feels pain. Thank you for your attention.
Thank you, Anna.